There is a waste disposal system inside your brain that science did not discover until 2012. Not because the technology wasn't there. Human brains had been studied for over a century by that point, dissected and imaged and analyzed with instruments of extraordinary precision. The system was present all along, in every brain that ever passed through a research laboratory, in every brain that ever belonged to a person who slept. The reason it took until 2012 to find it is that this system shuts down when you are awake. It activates only when you lose consciousness. And for most of the history of neuroscience, researchers were studying awake brains, or brains that had been chemically preserved in ways that collapsed the very microscopic channels they needed to see. The system was invisible not because it was small, but because it required a state that science had never thought to replicate in the laboratory. Nobody looked for a brain-cleaning system that ran at night, because nobody believed the brain needed one. What Maiken Nedergaard found in 2012, working at the University of Rochester with a team of neuroscientists and imaging engineers, changed the way we understand both sleep and the progressive neurodegenerative diseases that have become the defining medical catastrophe of aging populations worldwide. Her team's paper, published in the journal Science, described something that had been hiding in plain sight. A parallel plumbing system threaded through the entire brain, dormant during waking hours, activated by sleep, and responsible for flushing out the molecular garbage that accumulates during every hour you spend conscious. The magnitude of what she had found did not become fully clear until the field began mapping what happens to the brain when the system fails. She named it the glymphatic system. The name is a deliberate compound. Glial cells, the non-neuronal support cells that constitute roughly half the brain's total volume, and the lymphatic system, which performs analogous waste clearance functions throughout the rest of the body. The brain, uniquely among major organs, has no conventional lymphatic drainage vessels running through it. For decades, this absence was interpreted simply as a feature of neural architecture, a structural peculiarity with no particular consequence. The glymphatic system explains why that absence was never actually a problem. The brain built its own equivalent. It just runs only at night. To understand what this system does, you need to understand where it runs. The brain is bathed in cerebrospinal fluid, CSF, a clear, colorless liquid manufactured by structures called the choroid plexus, located within the brain's 4 fluid-filled chambers called ventricles. Your brain produces roughly 500 milliliters of CSF per day. The total volume present in the central nervous system at any one moment is only about 150 milliliters, which means the entire fluid reservoir turns over approximately 3 to 4 times every 24 hours. For most of neuroscience history, CSF was understood primarily as mechanical protection, a hydraulic shock absorber that cushioned the brain against the skull during movement and distributed intracranial pressure. What Nedergaard's team revealed is that CSF is also the medium of the brain's waste management infrastructure, pushed through tissue in a circuit that runs continuously during sleep and largely shuts down during waking. It flows not just around the brain, but through it, using a channel system that had been anatomically documented since the 1850s, but never assigned a function. Every blood vessel in the brain, every artery, arterial, and capillary, is surrounded by a narrow annular channel. A sheath of cerebrospinal fluid runs alongside each vessel, not inside it, but in the gap between the vessel wall and the surrounding brain tissue. This is the perivascular space, first described by Rudolf Virchow and Charles Philippe Robin independently in the 1850s and 1860s. These Virchow-Robin spaces were catalogued in thousands of histological preparations across a century and a half of neuroanatomy and consistently interpreted as inert anatomical features. In Nedergaard's model, they are the arterial input and venous output ports of a brain-wide hydraulic circuit. CSF enters the brain through the perivascular spaces surrounding arteries, moves through the neural tissue itself via specialized molecular channels embedded in a class of support cells called astrocytes, and exits through the perivascular spaces surrounding veins, ultimately draining into the lymphatic vessels running along the neck and through the cervical lymph nodes. The transit pathway through brain tissue passes through channels constructed from a protein called aquaporin 4, designated AQP4. These are water pores, molecular-scale portals embedded in cell membranes that allow water and small dissolved molecules to cross at rates far beyond what passive diffusion permits. Astrocytes extend long projections called endfeet that wrap around blood vessels in dense sheaths. These endfeet are loaded with AQP4 channels, and when cerebrospinal fluid moves through them, it carries in its current the metabolic waste products that neurons have released into the surrounding extracellular space: protein fragments, neurotransmitter metabolites, excess glutamate, oxidized lipids, and in particular 2 molecules now implicated in the most feared neurological diseases on Earth: amyloid beta and tau. The necessity of AQP4 in this circuit was demonstrated experimentally by Nedergaard's group using mice engineered to lack the protein. In AQP4 knockout mice, animals otherwise healthy, with intact brain anatomy and normal sleep behavior, glymphatic clearance of amyloid beta was reduced by approximately 65% relative to wild-type controls. The system did not simply slow down, it became functionally crippled. The fluid moved through the perivascular spaces but stalled at the astrocyte endfeet, unable to cross into the brain parenchyma at any meaningful rate. This single protein, AQP4, appears to be the rate-limiting bottleneck of the entire clearance circuit. Its distribution on astrocyte endfeet is not random. It is concentrated precisely at the interface between the perivascular space and the brain tissue, positioned exactly where convective flow must make the transition from channel to parenchyma. The spatial precision of that arrangement is not coincidental. It is the architecture of a system that was optimized over evolutionary time. And here is the mechanism that explains why the system is closed while you are awake. Norepinephrine, the neurotransmitter released by the locus coeruleus, a small nucleus in the brainstem that projects diffusely throughout the cortex, maintains the waking brain in a cellular volume state incompatible with glymphatic flow. Norepinephrine drives fluid into cells through beta-adrenergic signaling, maintaining the compressed extracellular space characteristic of arousal. When the locus coeruleus reduces its firing, which it does dramatically during non-REM deep sleep, falling to near zero discharge rates, norepinephrine levels in the cortex drop, and the cellular machinery holding cells at full volume finally releases them to contract. Bott and colleagues at Rochester demonstrated this by directly infusing norepinephrine into the brains of sleeping mice. Glymphatic flow dropped immediately. Blocking adrenergic receptors in waking animals produced a partial but measurable increase in interstitial space volume. The system is not merely activated by sleep, it is suppressed by wakefulness, through a specific neurotransmitter mechanism that evolution has coupled directly to the arousal state. But there is a second mechanism operating simultaneously, and this one is the reason sleep is not merely helpful, but structurally necessary. The glymphatic system does not operate uniformly across the 24-hour cycle. It activates during sleep, specifically during slow-wave deep sleep, and at a magnitude that Nedergaard's colleague Lulu Xie quantified directly in a 2013 science paper. Using 2-photon in vivo microscopy, which allows real-time visualization of fluorescently tagged tracer molecules moving through the living brain of an unanesthetized animal, Xie and colleagues measured the volume of the interstitial space, the gaps between cells, during sleep versus waking. The interstitial space increased by approximately 60% when the animals fell asleep. Not 5%, not 15%, 60%. The brain physically restructures itself at the cellular level the moment consciousness withdraws. The mechanism is cellular, and it is active, not passive. During wakefulness, neurons and glial cells maintain their full resting volume. When an animal enters slow-wave sleep, astrocytes and neurons actively reduce their size. They shrink in a controlled and reversible process driven by ion transporters in their membranes that move sodium, potassium, and chloride ions outward, pulling water with them by osmosis. The cells are not dying. They are deflating deliberately in a process that expands the interstitial channels surrounding them. And here the physics becomes decisive. In the narrow interstitial channels of the waking brain, fluid movement is governed primarily by diffusion, the slow, random Brownian motion of molecules migrating from regions of higher to lower concentration. Diffusion is inherently inefficient for clearing a tissue volume measured in centimeters when time is limited to a single night. The expanded channels of the sleeping brain support something categorically different: convective bulk flow, pressure-driven mass movement of fluid, carrying dissolved molecules along with it at rates orders of magnitude faster than diffusion alone permits. Sleep-induced cellular contraction converts a network of narrow slits into a high-throughput hydraulic system, purpose-built for rapid overnight clearance. The waste products carried by this flow are not incidental. Amyloid beta is a peptide fragment, between 36 and 43 amino acids in length, depending on which enzymes process it. That is a normal byproduct of neuronal activity. The amyloid precursor protein, APP, is processed continuously in active neurons, releasing amyloid beta fragments into the extracellular space as a routine consequence of synaptic signaling. Under conditions of healthy sleep, the glymphatic system clears these fragments every night, maintaining extracellular concentrations at non-toxic levels. When clearance fails, when glymphatic flow is insufficient relative to production, amyloid beta concentrations rise, small soluble oligomers form, then larger insoluble fibrils, then the extracellular plaques that define Alzheimer's pathology under the microscope. Whether plaque formation is a direct cause of neuronal death or a downstream consequence of earlier toxic oligomeric species remains scientifically debated. What is not debated is the correlation between insufficient glymphatic clearance and amyloid accumulation. Matthew Walker at the University of California, Berkeley, examined this relationship in human subjects across a series of studies published between 2017 and 2019. Using PET imaging with amyloid-specific radiotracer compounds and polysomnographic sleep monitoring, Walker's team demonstrated that reductions in slow-wave sleep amplitude predicted greater amyloid deposition in the medial prefrontal cortex over subsequent years. More directly, a 2017 paper from Walker's group showed that a single night of total sleep deprivation produced a measurable 5% increase in amyloid beta concentration in the cerebrospinal fluid of healthy human volunteers after 31 continuous waking hours. One night. 5%. The increase was most pronounced in the right hippocampus and the thalamus, the regions most centrally involved in memory consolidation and sleep regulation, which are also the regions most consistently affected in early Alzheimer's disease. Tau, the second molecular hallmark of Alzheimer's, follows the same clearance dynamics. Tau is a microtubule-associated protein normally found inside neurons, where it stabilizes the structural scaffolding that neurons use to maintain their form and transport organelles along their length. In Alzheimer's disease, tau becomes hyperphosphorylated, detaches from microtubules, and aggregates into the neurofibrillary tangles that progressively destroy neurons from within. Research from Nedergaard's group, published in 2019, showed that CSF tau concentrations follow a circadian rhythm, rising during wakefulness, falling during sleep, consistent with overnight glymphatic clearance. In their mouse models, chronic sleep restriction over 3 weeks produced tau aggregation pathology that was not fully reversed even after recovery sleep was permitted. The structural damage outlasted the deficit that caused it. The glymphatic system's failure extends beyond Alzheimer's in ways that are only beginning to be mapped. Traumatic brain injury, even mild concussion, disrupts glymphatic function through multiple simultaneous mechanisms. The physical trauma displaces astrocyte endfeet from their precise vascular positioning, disrupting the AQP 4-channel orientation that directs flow. Inflammation following injury causes astrocyte swelling, narrowing the interstitial channels the system depends on, and the pain, anxiety, and neurological disruption caused by the injury typically devastate slow-wave sleep architecture for weeks or months afterward, depriving the damaged brain of the clearance mechanism it needs most urgently to remove the cellular debris produced by the injury itself. Rog Aaron at Stanford and Nedergaard's group at Rochester published complementary findings in 2018 and 2019 showing that glymphatic clearance in rodent traumatic brain injury models was suppressed by 60 to 80% in the weeks following injury. This creates a collision of needs. The injured brain produces more molecular waste from dying cells than a healthy brain, precisely at the moment when the system for removing that waste is most impaired. Whether improving sleep quality after traumatic brain injury reduces the long-term risk of chronic traumatic encephalopathy is now an active clinical research question. Here is the part of the story I find most striking. Nedergaard's 2012 paper was not immediately embraced. The idea that cerebrospinal fluid moved through brain tissue via convective bulk flow was considered implausible by many established researchers. The brain's tight cellular architecture was thought to preclude directed bulk movement at any physiologically meaningful scale. The Virchow-Robin spaces had been documented for 150 years and assigned no function. The entrenched consensus held that the brain managed its waste locally, through lysosomal and proteasomal degradation within individual cells, supplemented by passive diffusion toward the ventricles. What Nedergaard's group did was place fluorescent tracer molecules into the CSF of living mice and watch them through a cranial window using 2-photon microscopy. The tracers moved, they followed arteries, they percolated through brain parenchyma along AQP4-rich astrocyte endfeet pathways. They emerged along veins and drained toward the neck. The movement was directional, sustained, and dramatically accelerated by the transition to slow-wave sleep. The data was not interpretable as diffusion. It was bulk flow, and it was visible in real time. When the technique was finally applied to this question, the answer required no interpretation. There is a clinical consequence to all of this that extends beyond any single diagnosis. Obstructive sleep apnea, the condition in which the airway repeatedly collapses during sleep, producing brief arousals that fragment slow-wave architecture, has been associated with accelerated cognitive decline at rates substantially above what age alone predicts. The mechanism proposed is glymphatic disruption. Each arousal terminates a slow-wave episode and resets the cellular expansion process. Nadia Gosselin at the University of Montreal published data in 2019, showing that adults with untreated obstructive sleep apnea had significantly elevated CSF amyloid beta and tau levels compared to controls, independent of age, sex, and ApoE genotype. Treating the sleep apnea reduced those biomarkers. The clearance machinery, given the opportunity to run, ran. What this suggests, carefully, because the human data is observational and the causal direction is not yet established, is that conditions which fragment slow-wave sleep may accelerate the molecular cascade toward neurodegeneration, and that treating those conditions may partially reverse it. And here is where sleep position becomes unexpectedly relevant. Jeff Iliff, a co-author on the 2012 paper, subsequently at Oregon Health and Science University, led a 2015 rodent study examining glymphatic transport efficiency as a function of body orientation. Lateral sleeping, lying on your side, produced more efficient amyloid clearance than the supine or prone positions. The proposed mechanism involves the geometry of perivascular drainage channels and the effect of gravity on CSF pressure gradients. Whether this translates directly to human sleep posture has not been controlled for in human studies. The authors were appropriately cautious about extrapolation. But the finding aligns with the fact that lateral sleeping is the most common spontaneous position among healthy adults globally, which may not be coincidental. What has been confirmed in human imaging is that glymphatic flow is coupled precisely to the electroencephalographic signature of deep sleep. Laura Lewis at Boston University, in a 2019 science paper using simultaneous fMRI and EEG during natural human sleep, directly imaged CSF pulsation in sleeping volunteers with high temporal resolution. The relationship she found was mechanistically specific. The large, slow oscillations of non-REM deep sleep, the 0.5 to 1 Hz waves visible on EEG as high-amplitude synchronized sweeps, were each followed by a measurable pulse of CSF flowing into the brain through the fourth ventricle. During the neural downstate of each slow wave, when cortical neurons fall collectively silent for a fraction of a second, blood volume in the cortex transiently decreases. This creates a pressure differential. CSF rushes in to fill the space. The cleansing is rhythmic, timed to neural oscillations. One pressure pulse per slow wave, repeated hundreds of times across a full night of deep sleep. Pull these mechanisms together, and the picture is complete. Every hour you spend awake, your neurons produce metabolic waste at a rate proportional to their activity. Amyloid beta fragments accumulate in the extracellular space. Tau cycles through synaptic terminals. Lactate, adenosine, and oxidized molecular species build up. The glymphatic system cannot efficiently clear these products during wakefulness because your brain cells are at full volume and the interstitial channels are too narrow for convective bulk flow. The system is structurally closed while you are conscious. When you fall into slow-wave sleep, your brain cells contract. The channels open. The slow-wave oscillations of your own neural activity drive pulsatile surges of cerebrospinal fluid through the expanded perivascular network, carrying the molecular residue of the day toward venous drainage and out of the skull. This cannot be compressed into a shorter time window. A nap captures some slow-wave sleep, but not the full architectural sweep of a night cycle. Alcohol suppresses slow-wave amplitude and reduces glymphatic flow even when total sleep duration is maintained. Aging reduces slow-wave amplitude by approximately 75% between the ages of 25 and 65. The AQP4 channels themselves change with age. Their precise polarization on astrocyte endfeet, the spatial clustering that makes directed flow possible rather than random leakage, becomes progressively disrupted. Bahat and colleagues at Rochester showed that aged mice have significantly reduced perivascular AQP4 density and correspondingly reduced clearance rates even when their slow-wave sleep architecture is pharmacologically restored. The machinery itself degrades. This means the same hours of sleep that adequately cleared the young brain may be genuinely insufficient for the older brain. Not because sleep has shortened, but because the molecular architecture of the clearance system has changed. The glymphatic system may be one mechanism through which biological aging is not merely correlated with cognitive decline, but mechanistically linked to it. At the beginning of this, you likely thought of sleep the way most people do, as absence. The body rests. The brain goes quiet. Consciousness pauses. What the glymphatic system reveals is something far more architecturally specific. Sleep is not the absence of neural activity. Slow-wave sleep is a particular and precisely organized form of neural activity, generating oscillations that serve as the hydraulic pump for a brain-wide clearance system. The quiet you experience as unconsciousness is, at another level of resolution, The sound of your own brain running its maintenance cycle. Cells contracting on schedule. Fluid surging through channels that were closed all day. Protein fragments being swept from tissue that produces them continuously and can only release them at night. Put your head down tonight. In perhaps 45 minutes, the slow waves will begin. Great synchronized tides sweeping across your cortex in the rhythm that Lewis measured and that Nedergaard's tracers followed. And with each wave, a pulse of clear fluid will move through channels that your own neurons have just widened to receive it. The molecular residue of everything you thought today, every problem considered, every image the visual cortex constructed and released, every conversation your prefrontal cortex processed and filed, will be carried out of the tissue, along venous walls, through the cervical lymph nodes, and away. It happens every night. It has happened every night of your life. Science learned it was happening 11 years ago.